Hello everyone, welcome to my channel. My name is Anubhab Laha. I am an assistant professor at Department of Botany, Chandanagar College. As a part of micro teaching of online refresher course in biological sciences being organized by UGC-Human Resource Development Center, Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad, I will be giving a short lecture on mutations. So in today's topic we will discuss about what is mutation, what are the different types of mutation, what are spontaneous and induced mutation, and what are the human disorders that can occur due to mutation. The term mutation actually refers to any change in the genetic material and also the process by which that change takes place. If that change takes place at a specific site in a gene, then it will be referred to as a point mutation, and it can affect the function of the genes, and thus this gene mutation can actually alter the phenotype by changing that function of a particular protein. Also, in some of the cases, the genetic mutation can occur when some genetic elements changes their position, like they move from one location to other, which can be seen in the case of transposable elements. Now coming to the types of mutation. The mutation is actually a process. As I have told earlier, there will be some changes in the base pair sequences of the DNA molecule. A cell which has a mutation can be regarded as a mutant cell. If it affects only a single base pair of DNA, then it will be known as base pair substitution mutation. These base pair substitution mutation and single base pair insertion or deletion mutations are actually different types of point mutation. So what about gene mutation? Gene mutation are those mutations which takes place in the sequence of the genes. This gene mutation can occur in a somatic cell or it can occur in a germline cell. But if it occurs in a somatic cell, that mutation will not be passed on to the succeeding generation. But if it takes place in the germline cell, then it may be transmitted by gametes to the next generation. So as I have told earlier, point mutation is of generally two types. One is base pair substitution mutation and the one is base pair insertion or deletion mutation. In case of base pair substitution mutation, there is a change from one base pair to another in the DNA. There are two types. One is transition, another one is transversion mutation. In case of the transition mutation, the change takes place from one purine pyrimidine base pair to other purine pyrimidine base pair. Like for here example, you can see that here AT gets changed into GC, where A and G are both purine and T and C are both pyrimidine. The next type is the transversion mutation, where the mutation causes a change of purine pyrimidine base pair to pyrimidine purine base pair, like the GC can get changed to CG or the AT can get changed to CG. Like here, this AT has been changed to GC in transition mutation, but when the transversion mutation is taking place, the CG is getting changed to GC. Now, the base pair substitution mutation can also be classified according to the effects which it can have on the amino acid sequence of the protein. The changes in the amino acid sequence in the protein can result in a protein which shows no change, or it can have an insignificant change or something a noticeable change. So coming to the first type of base pair substitution mutation is a missense mutation. In a missense mutation, the change in the mRNA codon is such that a completely different amino acid is inserted into the polypeptide. Like for example, here you can see the DNA is getting transcribed into mRNA, and this mRNA is getting translated into protein. This DNA sequence 5' AAA 3' is actually specifying lysine, which is a lysine amino acid in the protein. It gets changed into the 5' GAA, which actually specifies glutamic acid. So Due to this mutation, the AT gets changed into GC and it causes a missense mutation where a completely different amino acid, which is glutamic acid, is getting inserted instead of lysine. In case of nonsense mutation, however, this sort of change in the mRNA codon due to mutation can result in a stop or nonsense codon, which can be UAG, UAA or UGA. Here, for example, this 5' AAA 3' gets changed into 5' TAA 3', which when being transcribed into mRNA, it results in this particular UAA codon, which is a stop codon. So, as a result of this nonsense mutation, a premature termination of translation will take place. Thus, an incomplete polypeptide will be formed, because when there is a sense codon, then the corresponding tRNA carrying that new amino acid will be entering into the A site. But when this particular sense codon gets changed into a stop codon, like here, the release factor will come and it will result in the premature termination of the translation. In case of neutral mutation, however, the resulting amino acid substitution produces no such changes in the function of the protein. Why? Because the amino acid which is getting inserted after this particular change actually possesses the same property as like of the amino acid which was present before. Like here in this case, the 5' AAA 3' has been changed into 5' AGA 3'. Here lysine has been changed into arginine, but no detectable property change could be noticed because these both arginine and lysine are basic amino acids. Coming to the next type, which is the silent mutation, it is also known as synonymous mutation. Here, the altered codon in the mRNA specifies exactly the same amino acid in the protein. Thus, when this 5' AAA gets changed into 5' AAG 3', the same amino acid, that is lysine, gets inserted in the polypeptide chain, and this occurs at the third oval position of the codon, and this makes sense due to the degeneracy pattern of the genetic code. Another type of mutation is a frame shift mutation. In case of the frame shift mutation, there is a total shift in the reading frame downstream to the mutation. Like for here, if for some reason one or more base pairs are added or deleted, it can really alter the reading frame of the mRNA and can change the downstream of the mutation. Like for here, when there is an insertion of GC base pair before this particular lysine, as we know the codon are read in triplet like TCT, CAA, GAA, ATT. So after this glutamine, there was supposed to be lysine, then phenylalanine, but due to the insertion of the GC, a frame shift mutation takes place, and instead of this lysine phenylalanine, entire downstream the glutamic acid, isoleucine, tyrosine, etc. gets inserted. Thus, it generally results into a non-functional protein. And this frame shift mutation can also generate stop codon, which can result in a shorter polypeptide, or it can result in a longer than normal protein, or it can also cause significant alteration in the amino acid sequence of this polypeptide. There are also two more types of mutation according to their occurrence. Like it can be classified into spontaneous mutation if the mutations are occurring naturally, or it can be classified as an induced mutation where an organism is deliberately or accidentally exposed to a mutagen. Mutagen are the chemicals or the physical agent which can cause mutation in the DNA sequence of an organism. There are several diseases which can occur due to mutation. Like if for some reason a missense mutation takes place at the FGFR3 gene at the position 380, which can cause glycine to change into arginine, achondroplasia can take place. Like that, nonsense mutation, insertion mutation, deletion mutation can also cause several types of human disorders. A quite common disorder, which is cystic fibrosis, actually takes place due to a 3 base pair deletion of phenylalanine codon at the position 508 of the CFTR gene. Thus, coming to my conclusion, the mutations can be classified according to different criteria. They can be classified according to their cause, like it can be spontaneous or induced, their effect on the DNA, like it can be point or versus chromosomal, or it can be substitution versus insertion and deletion, or by their effect on an encoded protein, like it can be nonsense, missense, neutral, silent, or frame shift mutation. These are some of the references which I have consulted while making this PowerPoint. I hope you guys can get more information by going through these particular books. Thank you all. See you in next class.